So I've just presented my poster results at GI ASCO 2023. So my hypothesis for this study, entitled microRNAs as biomarkers of response to Trastuzumab in HER2-positive gastroesophageal cancer within the platform study, is that patients who are treated with Trastuzumab are generally other than HER2 positivity as a biomarker, we lack further biomarkers to enable us to predict response to therapy. So my study was looking at whether a particular predictive biomarker that I identified from my preclinical studies within my PhD might enable us to refine patient selection. So it was looking at the microRNA 148A3P. Our hypothesis was that patients with a high level of this microRNA might experience shorter overall survival, progression-free survival, and progression-free rates than patients with low microRNA levels. Using digital droplet PCR analysis, we initially had a sample of 63 patients, 41 of whom we were able to identify microRNA 148A3P levels through digital droplet PCR, and we were able to normalize 21 of these patient samples. We then looked to see whether there was any difference in survival using Kaplan-Meier survival curves, and we identified that patients with high microRNA 148A3P levels did not have any significant difference in overall survival or progression-free survival than patients with low microRNA 148A3P levels. However, we did find that there was a significant difference in progression-free rates at 3 months from commencement of maintenance Trastuzumab within the platform trial. The clinical context of this study is that patients who are HER2 positive are treated with induction chemotherapy and initial Trastuzumab alongside that chemotherapy, followed by maintenance Trastuzumab. So in particular, the maintenance aspect of that found that the progression-free rates at 3 months were different between these two microRNA cohorts. Further studies are required because it was difficult to normalize this microRNA to generate clinically meaningful results. We normalized it to microRNA 16, and there's lots of debate about how best to go about normalizing this to try and work out how to utilize this technology for patients. So further studies are ongoing.